Good afternoon, everyone. We will start today with a new seminar. The speaker is Mamata K C. She has been working from Auburn University in the USA. She has performed her master degree in plant pathology at this university under the supervision of Leonardo de la Fuente. So today she will present her master thesis that is entitled Silent Subgrowth and Biofilm Formation on Xylella fastidiosa and its connection with host adaptation and virulence. Just to mention that she just recently joined the University of Georgia as a PhD student in plant pathology. We are happy that she will continue her research on Xylella fastidiosa, specifically working in the pathosystem. I don't know in which crop, maybe grapevines, but she can explain later. So, Mamata, thank you for giving this seminar. The floor is yours.
Thank you so much for the introduction. Greetings, everyone. My name is Mamata K C. I recently completed my master's degree in plant pathology at Auburn University, and I'm currently a PhD student at the University of Georgia. Today, I'll be presenting my results from my master's research conducted at Auburn University under the guidance of Dr. Leonardo de la Fuente, where I focused on how xylem sap influences growth and biofilm formation of Xylella fastidiosa and how this relates to host adaptation and virulence. Let's start with the introduction. Xylella fastidiosa is one of the most challenging plant pathogenic bacteria due to its xylem limited nature and exceptionally broad host range. It can infect over 727 plant species worldwide, including many economically important crops such as grapevines, blueberries, almonds, olives, etc. The economic impact of Xylella fastidiosa is quite significant. For example, in California, alone, there occurs $104 million losses due to Pierce disease every year, and in Europe, where Xylella is one of the major emerging plant pathogen, it has been projected that losses might reach about 5.5 billion euros if pathogen spreads widely. Xylella fastidiosa is genetically diverse plant pathogen and is mainly divided into three primary subspecies: subspecies fastidiosa, subspecies multiplex, and subspecies pauca. These subspecies differ in geographic distribution as well as in host strains. For example, fastidiosa strains are mostly known for causing disease in grapevines and almonds. Multiplex strains are found to be associated with hosts such as blueberry, oak, and pauca strains are known for causing disease in olives. In addition to these main groups, two other in addition to these main groups, two other subspecies, Sandai and Morus, also have been proposed. Now moving on to geographical distribution of Xylella fastidiosa, it is now recognized as globally distributed pathogen, though it is native to America and was previously limited to continents of America. Now the pathogen is widespread and has become established in several parts of Europe and Middle East. Outbreaks caused in olives in Italy and almonds in Israel have caused severe economic losses there, and most recently, Xylella fastidiosa has been reported in Asia, including China and Taiwan as well. Altogether, this global distribution shows that it is adapting and spreading very fast across the world, which is kind of concerning for future disease outbreak. In plants, bacteria exclusively resides in xylem vessels, which are introduced by insect vectors. Once inside xylem vessels, the pathogen attaches to inner wall of xylem vessels and starts to form biofilm that eventually block the flow of water and minerals, and this blockage leads to water stress, which produces characteristic leaf spot symptoms. Biofilm formation is considered an important factor for virulence for this pathogen. In addition to biofilm, the plant also contributes to blockage through its own defense responses, for example, by forming tylosis, which is a balloon like outgrowth that can further block xylem vessels, enhancing the symptoms. Now, since we are talking about adaptation of this bacteria in host environment, something that we cannot leave out is xylem sap, because xylem sap is only the nutrient source for bacteria in plants, and it provides natural environment for bacteria. This that is why sap composition from different host plants can trigger different bacterial responses. Previous studies have shown that citrus xylem sap was able to inhibit biofilm formation of Xylella fastidiosa strains that are not pathogenic to citrus. However, the same strains were able to stimulate biofilm formation in grape sap and were known to be pathogenic in grapes. This difference here shows that biofilm is key factor for adaptation, and xylem sap has a definite role in adaptation of bacteria in plants. Now, while we understand how this bacteria causes infection in plants, the specific factors driving host specificity is still unknown, and host range is expanding every day. For example, blueberry infection, which was limited to subspecies multiplex, are recently found to be caused by subspecies fastidiosa as well. Similarly, some strains infecting grapes and almonds have shown cross-host pathogenicity infecting both plant species. These findings suggest that host strains boundaries are not strict for this pathogen, and adaptation of Xylella fastidiosa strains within xylem environment could be the reason for its expanding host strains. Thus, host strains experiments are very essential for predicting potential host sums, but they are often very time consuming and labor intensive. That's why identifying key factors is very important to understand host strains expansion. Now, building upon this knowledge gap, we wanted to ask, do Xylella fastidiosa growth patterns in xylem sap in vitro correspond to host plant adaptation? And to address this question, our central hypothesis is that the growth and biofilm formation of Xylella fastidiosa strains in xylem sap are predictive of their virulence and symptom expression in planta. The general objective of this study is to understand how xylem sap environment influences bacterial adaptation and host interactions, and specifically, we aim to first evaluate the growth and biofilm formation of multiple strains in both host and non-host sap, and also assess adaptive capacity of few selected strains, and second, link those in vitro traits to symptom development in grapevines and blueberry plants under greenhouse conditions. To test this hypothesis, we designed experiments using two host systems, Grapevine and Blueberry. Grapevine being a classical host for Xylella fastidiosa and Blueberry being an emerging host. For both the hosts, I performed two complementary assays. In vitro assays, where I assessed growth and biofilm formation of multiple Xylella fastidiosa strains in xylem sap collected from each host, and in planta assays, where I evaluated disease severity and bacterial colonization under greenhouse conditions. Now, first, I will talk about Grapevine host system. One of the reasons for choosing Grapevine system for our experiment is because Grapevine being one of the highest value crop in US, as the impact of wine industry in US economy is $320 billion per year. Another reason is that it is a well characterized host for Xylella fastidiosa causing Pierce's disease. As I previously stated, there has been reported cross-host pathogenicity between grapevines and almond strains, and these strains are also found genetically closer. Thus, testing a diverse set of Xylella fastidiosa strains, including almond isolated strains in grapevines, is crucial to understand cross-host compatibility, and also grapevine sap provides a natural environment to test strain behavior. For the in vitro experiments with grape sap, I mostly used xylem sap extracted from susceptible variety Chardonnay. And few experiments were also done using resistant variety Blanc du Bois. The sap was collected directly from freshly cut grapevine canes during early spring, where when the xylem flow is very active. Then we further filter sterilized xylem sap and used it for our experiments. For in vitro assays with susceptible sap, we used PD3 media as base growth medium and then supplemented it with grapevine sap at 25, 50, and 75% concentration. We only use 25% SAP amended media in case of resistant SAP experiments due to limited SAP availability from resistant variety. To strengthen our study, we used a diverse collection of Xylella fastidiosa strains representing different hosts, subspecies, and geographic origin. As you can see here, our collection includes strains isolated from grapevines, almonds, and blueberry, which are major hosts associated with the disease. These strains cover major subspecies fastidiosa and multiplex. And geographically, they represent isolates from California, Florida, Georgia in the US, as well as from Spain and Israel. Now, all these diverse strains were used for our in vitro assay. They were grown in PD3 media and PD3 media amended with grapevine sap at 25, 50, and 75% concentration. The bacterial cultures were adjusted to zero point 0.2 OD and inoculated into 96 well plates containing respective media. The plates were incubated at 28 degrees Celsius for 7 days, and growth was measured every day at 600 nanometer. And at the end of the experiment, biofilm formation was quantified using 0.1% crystal violet. Now, this is the result for biofilm formation by grapevine isolated strains in grapevine sap, and they all belonged to subspecies Fastidiosa. Here in x-axis, it represents different media treatments, and y-axis represents the transformed biofilm um, optical density value measured at 600 nanometer, which indicates the amount of biofilm produced after 7 days of incubation. For the three strains here, we observed a clear and significant increase in biofilm formation as the concentration of grapevine sap increased, whereas one of the grapevine isolated strain showed relatively low biofilm formation with only modest increase at higher sap concentration. Similarly, here is the result for the strains that were isolated from almonds, and they also belong to subspecies Fastidiosa. Four of the strains here followed similar trend as grapevine isolated strains with increase in biofilm formation when sap concentration was increased. The remaining two strains showed modest increase in biofilm formation with increase in percentage of sap. Now, moving on to the results from subspecies multiplex within almond-derived strains. Compared to the fastidiosa group we just saw, these multiplex strains generally produced much lower biofilm in grapevine sap. Four of the strains here, they produce very low biofilm even at high sap concentration. Two strains here showed modest increase in biofilm at high sap concentration, and one of the strain showed high biofilm at 50 and 75% sap. Now, we also did the same experiment for two of the blueberry isolated strains, and biofilm formation for both of these strains increased significantly when the percentage of sap was increased. Now, after these in vitro assays, we wanted to know if the in vitro well plate results hold true in plant as well. So, to complement the plant-based biofilm assays, we used a microfluidic chamber system. Because this system mimics plant xylem vessels, it allows us to observe real-time bacterial behavior under flow conditions. And here, two different media is passed through these media inlets, In our case, we used 50% Grapevine sap, amended media, and PD3 media, and the bacterial suspensions were passed through these bacterial inlets, and this chamber allow us to continuously pass media through these two channels while directly observing bacterial behavior under microscope. For this experiment, two almond isolated strains with contrasting phenotype in our well plate assay was selected. Strain ALS 17T10, which formed a strong biofilm in sap, and strain Dixon, which did not form much biofilm in sap, in our 96 well plate assay. So this is what we observed. The top channel here is sap amended media, whereas the bottom channel indicates PD3 media for both of the strains here. And the strain here, on your left, you can see the strain that is forming substantial amount of biofilm in sap amended media as compared to PD3 media, whereas the strain on right, it also has enough cells, but it is not able to form biofilm in sap amended media, and it is showing a clear difference here. And this observation under flow condition further validated our plate results. After observing these results of biofilm formation in vitro and microfluidic condition, we wanted to determine whether these differences of biofilm actually translate into disease development inside plants. So we conducted in-planta inoculations using susceptible grapevine variety Chardonnay. For in-planta inoculation, each strain was grown to 0.3 OD, and 20 microliters of bacterial suspension was inoculated at the base of the plants. A needle prick method was used for inoculation. Following the inoculation, plants were maintained under greenhouse condition and monitored closely for disease symptoms progress. At the end of experiment, we measured bacterial colonization using quantitative real-time PCR to assess bacterial population within plant tissue. Now this slide here shows how symptoms gradually develop in grapevines infected by Xylella fastidiosa. The disease starts with marginal leaf necrosis as in here, which progresses inward, and many leaves dry completely and fall off, leaving behind only the petioles, leading to characteristic matchstick symptom. In severe stages, almost all the leaves become scorched and infection leads to death of the vine. Now here is the result for our in-planta experiments. The graph here shows disease progress in grapevines inoculated with diverse Xylella fastidiosa strains over 9 weeks. Y-axis here represents disease severity index and x-axis represents number of weeks after first symptom appeared. As you can see, strong strains belonging to subspecies fastidiosa shown in the upper group caused rapid and severe symptom development, reaching over 50% disease severity index by week 9, whereas most of the strains from subspecies multiplex shown in lower group induced mild symptoms with disease severity index generally below 25%. Furthermore, these two graphs here summarize overall disease intensity and bacterial colonization results from grapevine in planta experiments. The graph on your left represents disease under area under disease progress curve with strains on x-axis and average AUDPC on y-axis, whereas the graph on your right represents bacterial population with strains on x-axis and log CFU per milligram petiole on y-axis. Here also the strains belonging to subspecies fastidiosa showed significantly high disease intensity compared to the strains belonging to subspecies multiplex. Similarly, the results correlated with bacterial population as well, as fastidiosa strains showed significantly high bacterial load compared to subspecies multiplex strains. These results indicated that subspecies fastidiosa was better adapted to grapevines and has potential to cause severe disease in grapevines than subspecies multiplex. Now here is the summary that shows only the strains belonging to subspecies fastidiosa was able to cause disease in grapevines, whereas strains that belong to subspecies multiplex, they were not able to cause disease. And our hypothesis was that biofilm in sap could predict symptoms in plants. So strains in the top two boxes supported our hypothesis, whereas the strains in the bottom two boxes, they did not. And overall, to summarize, most of the strains, biofilm formation, for most of these strains, biofilm formation in vitro correlated to symptoms development. Now, after seeing some correlation in biofilm formation and symptom development, we wanted to see the effect of sap from resistant cultivar on biofilm formation. So we tested a few virulent strains isolated from grapevines in resistant sap and susceptible sap for biofilm, and we observed that uh, the strains form significantly less biofilm in sap from resistant cultivar than the sap from susceptible cultivar, and this result suggests that resistant cultivar may limit the infection by creating less favorable environment for biofilm formation. Now, after all these in vitro microfluidics and in planta experiments, we saw that biofilm formation is a very important factor for adaptation of bacteria, and we also saw differences in biofilm as well as in virulence in different strains. So we wanted to do a preliminary experiment to investigate the adaptation potential of Xylella fastidiosa in grapevine sap. um, so we already know that Xylella fastidiosa is a naturally competent bacteria. That means that it can take DNA from environment and incorporate it into its genome by homologous recombination. So based on our results and this information, we asked, can a strain that is not adapted to non-host xylem sap can take DNA from environment to adapt in that particular xylem sap? So to do that, we focused on two of the strains. They were isolated from almond, and they differ in their biofilm phenotype as well as virulence in plant. So we selected strain ALS 17 T10, which formed high biofilm in grapevine sap and was virulent in grapevines, and ALS 6 that formed less biofilm and was avirulent. So to track each of the strains were marked using different antibiotic resistant cassette inserted at NS1 non-coding region of the genome, and these strains were either grown alone in sap amended media, or dead cells of the other strain was added to facilitate natural competence and adaptation. And this was our experimental setup. I conducted this experiment in 24 well plate. First, I inoculated respective bacterial suspension in respective media. After 7 days, I measured the total bacterial growth and then transferred the culture to new plate by mixing vigorously. For new plate, I used 80% of fresh media and 20% of old culture, and it was again grown for another 7 days, and another passage was done repeating the same process, and readings were taken before and after the passage was done both the times. And here is the result for serial passage experiment. Here you can see the strain ALS 17 T10 that formed high biofilm in sap and virulent in plants was able to adapt more and more in sap amended media when it reached 21 days, compared to the same strain in PD3 media, whereas the strain ALS6 was that was avirulent in plants and formed less biofilm in sap, did not grow well when we did the passes every week, and by day 21, the strain did not survive in sap amended media. We did the same experiment again, but this time we also added dead cells for alternate bacteria alternate bacteria to see if bacteria would take DNA from dead cells to incorporate genes required for adaptation under selective pressure. But unfortunately, we did not observe any significant difference even after the addition of dead cells. Now, to summarize all the findings from grapevine systems, strains belonging to subspecies fastidiosa clearly showed strong adaptation to grapevine. They produced severe symptoms, reached high bacterial loads, and many formed significantly more biofilm when they were grown in grapevine sap. Um, Interestingly, a few virulent strains did not respond to sap with more biofilm, indicating that they may rely on other mechanisms such as rapid systemic spread rather than strong attachment. In contrast, the multiplex strains mostly showed poor adaptation to grapevine. They showed low symptom development and colonization, and even when they did form some biofilm in sap, they did not translate it to virulence. We also saw highly virulent strains where not able to produce significant biofilm in resistant sap compared to susceptible sap. And finally, the serial passes experiment also showed that virulent strains was much more adapted to sap through multiple passes. Now, after looking at the results in grapevines, we also wanted to see if similar host specific patterns could be seen in blueberry host system. So, for blueberry, also both in vitro and in planta assays were done. Mhm. The reason for studying blueberry was because blueberry, particularly hibiscus cultivars here in US, is economically very important. Mm, and if you look at the graph here, red bar shows the increase in blueberry production in southern states over years, and its cultivation is now expanding rapidly across Europe as well. And as I previously stated, that the infection of blueberry is not limited to subspecies multiplex, and it raises concern about the host strain's plasticity of Xylella fastidiosa. And um, we thought, what if other subspecies also can infect blueberry? Since blueberry is an emerging host for Xylella fastidiosa, its production is also increasing, and this host system has not been explored very well. This makes blueberry more vulnerable host to diverse Xylella fastidiosa strains. So we thought that it is essential to test different strains of Xylella from different subspecies and host origins to reveal cross-host compatibility and adaptation of this pathogen in blueberry as well. So for our in vitro experiments, unlike grapevines, it was very difficult to collect sap from blueberry plants. I started growing some plants in greenhouse to collect sap. I could only get around 1 to 1.5 milliliter of sap from a single fully grown plant in 1 gallon pot. I used negative pressure xylem extraction unit for sap collection, and mm, I did most of the experiments in susceptible sap and few of the experiments in resistant sap. Now again for blueberry also, we use different diverse collection of Xylella fastidiosa strains representing different hosts, subspecies and geographic origins. As you can see here, the collection includes strains belonging to 4 subspecies and 7 different hosts, and geographically they represent isolates from different parts of US, Costa Rica, Mexico, Italy, Spain, and Israel. The in vitro setup for blueberry sap was also similar as in grapevine sap, but we were only able to use 25% of blueberry sap due to limited amount of sap present. Now moving on to the results from in vitro experiments, starting with the results from subspecies multiplex. Two strains here, mm, they showed significantly in higher biofilm formation in blueberry sap as compared to PD3 media, whereas the other three strains that belong to subspecies multiplex, they showed negative response. Though blueberry subspecies multiplex are known to cause disease in blueberry plants, and we had only one of the strains from subspecies sandai, and it also showed decreased biofilm response in sap. And this is the result for subspecies fastidiosa. And subspecies fastidiosa were thought to be virulent in blueberry, but interestingly, we saw um, most of the strains had significantly higher biofilm in sap amended media as compared to PD3 media, and two of the strains showed decrease in biofilm formation in sap as compared to PD3 media. Now, after these in vitro assays, we again moved on to in planta experiments to test whether these differences we observed in biofilm formation would translate to differences in virulence and colonization in blueberry plants as well. So, similar to grapevine in planta experiment, we inoculated diverse set of strains representing four subspecies in susceptible cultivar of blueberry, and we monitored symptoms and also quantified bacterial colonization using qPCR. And here is the result for Implanta assay. The graph here represents disease progress curve for strains inoculated in blueberry. We observed that severity of symptoms among these strains and subspecies varied very widely in blueberry plants. Strain Alma EM3, which was our positive control, showed the most severe and consistent symptom development, reaching more than 90% by week 9. Similarly, the other group of strains also produced severe symptoms in blueberry, reaching almost 75% severity by week 9. And for the third group here, we observed mild symptoms for some strains, and their disease severity index was less than 50%, and some of them were asymptomatic as well. Now, based on the disease severity index, overall disease intensity across experiment was also calculated, consistent with previous disease progress curves. Strain Alma EM3 caused highest disease severity, significantly greater than all other strains. The second group here also showed high UDPC values, indicating severe symptom development, and the third remained mild to nearly asymptomatic in blueberry plants with low ADPC values. Now let's look at the result for bacterial colonization in plants. Here mostly strains showing high disease intensity also maintained high bacterial population. However, there were some of the strains which had high bacterial population but caused relatively mild or minimal symptoms, showing that certain strains can establish well in host without necessarily inducing severe disease. This slide here shows the overall summary of Implanta experiment in blueberry. Here you can see, unlike grapes, where only subspecies fastidiosa was able to cause disease in grapes, but in blueberry, not all multiplex strains causes disease, which were known to be causing disease in blueberry, and here many of subspecies fastidiosa strains are also able to cause disease, which were previously known to be asymptomatic in blueberry. And to our surprise, even one of the strain from subspecies pauca caused severe disease in blueberry and had enough bacterial population in plants. Before this, it was not reported that subspecies pauca can cause disease in blueberry, which is interesting also and also kind of concerning because the strain was isolated from lavender, which is a kind of some weed. That means that some weeds can be serving as reservoir for such virulent strain, and if such strains get transmitted by vectors in susceptible host such as blueberry, it has potential for causing another severe outbreak in blueberry or some other susceptible host. So again, I wanted to correlate my in vitro results to in planta results here. And here, most of the strains forming high biofilm in sap amended media showed severe symptoms in plants, whereas strains forming less biofilm in sap amended media were mostly asymptomatic. And two of the strains did not follow our hypothesis. Now, as in grapevines, blueberry also I wanted to test few blueberry isolated strains in resistant and susceptible sap, and I observed that in blueberry also biofilm formation decreased significantly in resistant sap as compared to susceptible sap, indicating that resistant variety could be limiting infection by creating unfavorable environment for biofilm formation. Now, to summarize blueberry host system, mostly we observed strain-specific adaptation in blueberry irrespective to subspecies and host. And um, one of the strain from subspecies Pauca was able to cause severe disease in blueberry. Similarly, biofilm formation also is strongly correlated with virulence in plants, and biofilm formation decreased in resistant sap as compared to susceptible sap. Now, to summarize overall results from my master's thesis, our findings shows that virulence and adaptation in Xylella fastidiosa are complex and highly strain-specific process, and they cannot be only explained by subspecies identity, which means two strains from same subspecies can behave very differently depending on host or environment. Secondly, the detection of new virulent strains in blueberry highlights the possibility of cross-host infection. This suggests that Xylella population in field are dynamic and thus continuous monitoring of strain diversity is necessary. Our results also emphasize both the xylem environment and bacterial traits jointly shape disease outcomes. Therefore, a deeper understanding of host pathogen interaction is important to predict host germ and limit spread of Xylella fastidiosa. And finally, xylem sap mediated biofilm formation could serve as a rapid and early stage screening tool for evaluating strain virulence and host adaptation potential before any greenhouse or field trials, which take a lot of time. Now, before wrapping up, I would also like to briefly mention some limitations. First, the limited availability of blueberry sap restricted the number of concentration that we could test, and because of that, we might have underestimated the dose-dependent effects. Secondly, while our most of the plate-based and flow-based assays were very informative in understanding bacterial behavior, they still cannot fully replicate the complex biological interaction that occur mm, within living plants because of plant defenses and other microbial competitions. And finally, our greenhouse experiments were conducted under controlled conditions, and we used mechanical inoculation, which is different from field infection transmitted by insect vectors. With that, I would like to thank my major advisor, Dr. Leonardo de la Fuente, for his continuous advice and motivation. I'd also like to thank my committee member, Dr. Neha Portnes and Dr. Jonathan Oliver. I'd like to thank all of my past lab members and current lab members. And I'd also like to thank Jessica, Associate Director from Greenhouse, who helped me a lot in greenhouse essays. And finally, I would like to extend my gratitude to funding agencies, Bexit Project and BART Project. And thank you very much for listening. And I'll take any questions. Thank you.
Thank you a lot, Mamata, for this clear presentation. So is there anyone that wants to make questions to Mamata?